Welcome to my channel. Topic of discussion is evasion of human immune system by pathogenic bacteria. Pathogenic bacteria are known to have evolved several strategies for escaping or evading human immune system. The first strategy employed by these pathogenic bacteria is the penetration of the skin. The intact skin or the unbroken skin is known to provide an effective mechanical barrier against pathogens, which are disease-causing microorganisms. However, if there's a break in the skin, such as a burn or wound or insect bite or medical procedure such as insertion of a catheter, these may provide direct access of microorganisms to tissues below the skin. Staphylococcus epidermidis is a member of the normal flora of the intact skin, where it is regarded as a non-pathogen, that is, it does not produce disease. However, if there is a break in the skin or a breach in the intact skin as provided by a wound or a burn, Staphylococcus epidermidis may find its way to tissues below the skin. Particularly, it goes straight to the heart where it causes endocarditis. The second strategy used by pathogenic bacteria is the infiltration of the mucous membranes. The mucous membranes line the various systems of the body, such as the respiratory system, the urogenital system, the conjunctiva, the gastrointestinal tract. The mucous membranes produce mucus, which is made up of glycoproteins. Now, the function of the mucus or the mucin layer is to trap pathogens, which are then removed by phagocytes in the process of phagocytosis. However, some pathogenic bacteria lack surface proteins or carbohydrates that are required to bind these mucin components. So they are able to penetrate the mucin layer without being trapped. Another important feature is that the mucin layer creates a buffer zone with neutral pH. This has been taken as an important safe hiding place for intestinal bacterial pathogens, such as Helicobacter pylori, which causes peptic ulcer. The M cells of the gastrointestinal tract are known to be targets of several pathogenic bacteria that cause intestinal diseases. The M cells have large pockets containing macrophages, B cells, and T cells. The M cells are known to periodically sample the content of the gut lumen. More or less, they serve as police officers. And so they interact with pathogens which may be present therein. And with the aid of the macrophages, the pathogens are eliminated in the process of phagocytosis. The B cells that are found are transformed into plasma cells. These plasma cells in turn produce secretory IgA, which interact with the pathogens in the gut lumen. It is important to note that some pathogenic enteric bacteria do not degrade mucin actively, and so they may use this entry portal into the bloodstream if they are able to resist the destruction by the M cells. The third strategy used by pathogenic bacteria is resistance to antibacterial peptides. What are these antibacterial peptides? They are known as defensins. They are cationic in nature, that is, they are positively charged. So all the pathogenic bacteria requires is to identify a component in their cell wall and use it against them. For example, gram-negative bacteria are protected by their lipopolysaccharides, which are negatively charged. And so they bind to the cationic peptides. Another defense strategy is that peptidases, which destroy the defensins, are produced even before they arrive at the cytoplasmic membranes. The fourth important strategy used by pathogenic bacteria for evading the immune system of humans is the production of secretory IgA proteases. Secretory IgA is responsible for the slimy nature of the mucin layer, and they normally bind to pathogenic bacteria through the antigen binding sites, and they bind to the mucin layer through the Fc portion. The secretory IgA proteases are extracellular enzymes which are produced by these pathogenic bacteria that cleave human IgA in the hinge region, thereby rendering them ineffective. Ion acquisition mechanisms have also been used to evade the human immune system. Iron is sequestered into ion compounds such as hemoglobin, lactoferrin, and thereby make iron unavailable to pathogenic bacteria for their metabolism. Well, pathogenic bacteria will have to carry on their activities and they have to source for this iron. So they develop some molecular structures known as siderophores to chelate iron for their use. Yet another important ion acquisition strategy of pathogenic bacteria is simply to produce toxins. These toxins will just kill the host cells when the iron level is low. And once the host cells are killed, the sequestered ion compounds will be made available to the pathogenic bacteria for their use. Another important strategy used by pathogenic bacteria is the production of invasins. These invasins are bacterial surface proteins. These initiate or cause cytoskeletal rearrangement in normal non-phagocytic cells, such as the host epithelial cells. So they escape phagocytosis by entering these host epithelial cells, and they are protected. Yet another strategy used by pathogenic bacteria is the prevention of phagocyte migration. C5a is a component of activated complement system. The C5a has the function of causing phagocytes to migrate to the site of infection. So it has been discovered that some gram-positive cocci produce a bacterial enzyme which is known to degrade C5a. And so the function of causing migration of phagocytes is lost. This action also reduces the oxidative burst in which reactive oxygen intermediates are released for the intracellular killing of any engulfed pathogen. Another important strategy used to evade human immune system is escape from phagosome. Some pathogenic bacteria we just watch. We don't care if you like what loss of in the membrane-bound structures known as phagosome. We'll do something. So they produce proteins that degrade membrane lipids or form pores in the membrane of phagosomes. And so they escape from the membrane-bound structure known as phagosome. Another important strategy used by pathogenic bacteria is prevention of phagolysosome formation. Well, the pathogen is in a membrane-bound structure known as phagosome, and the next stage will involve the fusion of the lysosome granule to this phagosome. Some pathogenic bacteria are known to prevent this fusion of the lysosome granule to the phagosome, thus preventing the killing of the bacterium that is engulfed. Prevention of complement activation is also another strategy that is used by some pathogens. They protect against complement activation and killing by phagosomes. This they do by preventing the formation of the C3 convertase.